for the introduction and for allowing me to share my research. I'm excited to tell you all a research story today about the identification of intra-Golgi transport pathways. To begin, I just want to orient all of you to the position of the Golgi apparatus. I like to say that it lies at the heart of the endomembrane system. It's comprised of multiple cisterna, and these cisterna collectively perform two primary functions for the cell. The first being the enzymatic modification of proteins and lipids, and the second, they are sorting and export to other organelles or the cell exterior. To perform these critical functions, the Golgi apparatus polarizes its own proteins across its stack of cisterna. Here, I hope you can appreciate from this data obtained by Lei Lu's group that each resident Golgi enzyme or trafficking machinery protein has a center mass of distribution in the context of the Golgi stack. Golgi polarization is an evolutionarily conserved feature of the organelle, and it ensures that cargo proteins that traverse the Golgi can first undergo enzymatic modification before sorting and export. However, the core functions of the Golgi, namely the anterograde transport and sorting of proteins, actually poses a very interesting challenge for the organelle. And that is simply, how can the Golgi retain and polarize its own proteins in the face of this membrane flow in the anterograde direction? In the Glick lab, we believe the cisternal maturation model describes how the Golgi can retain and polarize its own proteins. This animation on the right depicts the model, which essentially postulates that each individual cisterna undergoes a life cycle of formation, maturation, and disintegration. At the bottom, you see cisternal formation occurring, and this is essentially when COP2 vesicles budding from the ER homotypically fuse, and this is when secretory cargo import occurs. In the middle, each cisterna matures by receiving resident Golgi proteins from older cisterna in vesicles. And at the top, you can see the final stage where the cisterna disintegrates into different carriers for protein sorting and export. Now, the maturation model states that secretory cargoes largely remain within cisterna as they mature. However, the resident Golgi proteins themselves must recycle from older to younger cisterna using specific retrograde vesicular intra-Golgi transport pathways. These pathways have been thought to exist in the field for many years now, but they've not actually been molecularly identified and defined. And this is the objective of the work that I'm going to share today. So to understand and define these pathways, we actually need to look at the localization of resident Golgi proteins in detail, but that's tricky in most eukaryotic cell types because the Golgi is usually stacked, which makes it impossible to cleanly resolve individual cisterna by light microscopy. This is why we study the Golgi in budding yeast. Budding yeast have unstacked cisterna that can be individually resolved, and when we perform time-lapse imaging of individual yeast Golgi cisterna, we observe that Golgi proteins sequentially arrive and depart over time. This is not only consistent with the maturation model, it underscores the fact that Golgi polarization is actually a temporal phenomenon from the perspective of a single cisterna, whereas from the perspective of a maturing stack of cisterna, you could think of it as a spatial phenomenon. When we graph this temporal localization over time, we obtain what we call kinetic traces for different resident Golgi proteins of interest. These two in the example here are transmembrane proteins, so they are proteins that would be recycling in vesicles. When we obtain enough of these individual kinetic traces, we can actually average them and obtain definitive kinetic signatures for any given resident Golgi protein of interest. Now, our kinetic studies in budding yeast have revealed essentially multiple distinct kinetic signatures for transmembrane resident Golgi proteins. And you can see 3 of them here in this diagram, which I've labeled early, intermediate, and late. And the x-axis here simply represents the cisternal maturation timeline. So we can understand, we can suppose that each of these traces may represent a distinct intra-Golgi pathway, and therefore a distinct vesicle population. If this understanding of intra-Golgi transport and these kinetic signatures is accurate, then we can use this animation to understand what's happening at the level of the vesicle. Here, we could say that the first half of a protein's kinetic trace is its arrival phase. During this time, vesicles with the protein of interest are fusing with the cisterna. And then the second half of the trace could be supposed to be its departure phase, where vesicles with the protein are budding and departing from the cisterna. If this understanding is accurate, then we can make the prediction that only transmembrane Golgi proteins with identical kinetic signatures should traffic in vesicles together. And it's this understanding that we decided to test. And to do so, we developed an in vivo vesicle capture assay. So this assay allows us to determine whether two proteins traffic in vesicles together. In the diagram, you can see that our assay uses the rapamycin-inducible heterodimerization system to capture and concentrate a population of vesicles at the yeast septin ring. This population is defined by the presence of an FRB domain tagged resident Golgi transmembrane protein. So this allows us to capture vesicles only with this protein. To perform the assay, we GFP tag a second protein, here shown in purple, and we ask whether that protein appears at the septin ring when we capture the specified vesicle population. If it does, then we can conclude that the two proteins, the two tag proteins here, are present in the same vesicle population. So to begin, we ask whether proteins with the same late kinetic signature, here shown in the diagram, traffic in vesicles together. And we chose the late Golgi protein KEX2 to define this vesicle population. And we asked whether another protein sharing the same kinetic signature as KEX2 is also present in vesicles. And I hope you can appreciate from this image that rapamycin addition causes a sterol13 to colocalize with the yeast septin ring, but not the Golgi cisterna at that location. And what this tells us is that sterol13 is present with KEX2 in vesicles that have been captured at the septin ring specifically. We can represent this result quantitatively as a fraction of total cellular sterol13 signal that is colocalizes with the septin ring, but not the Golgi. In addition to sterol13, we found another protein, STV1, that has the late kinetic signature and is also present in vesicles with KEX2. However, an early Golgi protein, VRG4, is largely absent. So these data, at least preliminarily, support our hypothesis that different kinetic signatures represent distinct vesicle populations with regard to cargo composition, and therefore could also be said to represent unique intra-Golgi transport pathways. Next, we asked whether proteins with the same early kinetic signature also traffic in vesicles together. We chose the early Golgi protein VRG4 as a hook for vesicle capture, and we found two other early Golgi proteins that also reside in vesicles with KEX2, or excuse me, with VRG4, but the late Golgi protein KEX2 is not present in these vesicles. So this is also consistent with our hypothesis regarding the interpretation of individual kinetic signatures as distinct transport pathways. Finally, we investigated proteins with the intermediate kinetic signature. We were intrigued to find that we could not detect substantial amounts of two intermediate proteins in the early and late vesicle populations that I just defined for you. And this result excited us because it suggested that a third vesicle population enriched with intermediate proteins exists in the cell. However, it was difficult for us to assay this population because we were unable to cytosolically FRB domain tag an intermediate protein without perturbing its localization. Fortunately, we found another protein, cis1, that actually appears to partition between the late trafficking pathway and the putative intermediate pathway. And when we captured vesicles with the cis1 protein at the septin ring, we observed substantial amounts of the same two intermediate proteins. So what this tells us is that a vesicle population enriched with intermediate proteins does exist, and we believe that this vesicle population is represented by the intermediate kinetic signature, and is therefore a unique trafficking pathway. So we can sum up our results here in this kinetic diagram. We believe that we've identified three distinct intra-Golgi transport pathways with different vesicle populations that occur at early, intermediate, and late stages of cisternal maturation. But we were interested in further defining these pathways using the machinery that actually operates each one. As I mentioned before, the protein cis1 seems to partition between two pathways. And so it became apparent to us that it's not actually the most accurate using cargo composition to define a pathway. It's just one piece of the puzzle. Now, we know from the literature that COP1 is the coat that forms the vesicles of the early pathway. And we more recently in our lab published evidence that the clathrin coat and the AP1 and N5 clathrin adapters form the vesicles of the late pathway. However, in our present study, we had just identified the intermediate pathway, and we did not know what coat and adapter proteins formed its vesicles. So we began by asking whether the intermediate pathway uses the AP1 and N5 clathrin adapters. It's possible that a single pathway, that two different pathways could use some of the same machinery. So to answer this question, we used an assay that we've previously published that tests for the dependence for a protein's dependence on AP1 and N5 to be recycled within the Golgi. When we delete the AP1 and N5 adapters, we cause proteins dependent on them to be secreted to the plasma membrane, where they're then endocytosed and returned to the Golgi. In the context of our assay, what we do is we add the drug CK666, which inhibits endocytosis, and this causes protein dependent on AP1 and N5 to accumulate at sites of polarized secretion in the context of the AP1 and N5 double mutant. So we decided to test these four proteins, KEX2 sterol13 from the late pathway, TMN1 from the intermediate pathway, and VRG4 from the early pathway. We used the assay to test them for AP1 and N5 dependence. And here I'm showing you the data. I hope you can appreciate here that both KEX2 and sterol13, we observe them at sites of polarized secretion in the double mutant context when we add the drug CK666 to inhibit endocytosis. However, we did not see VRG4 and TMN1 at these sites of secretion under the same conditions. So what this tells us is that both the early and intermediate pathways are not dependent on AP1 and N5, whereas the late pathway is dependent, consistent with our previously published data. In addition to this assay, we performed some partial kinetic analysis of the intermediate pathway TMN1 against a subunit of the AP1 adapter complex. And essentially what I found was that I could barely detect overlap between the two proteins. It seems that TMN1 is largely departed from maturing cisterna by the time AP1 arrives. And this basically tells us that AP1 is not even present at the Golgi to be largely responsible for TMN1 departure. So on top of the functional data, the kinetic data also support a picture where the intermediate pathway does not actually rely on AP1 and N5 to form its vesicles. So we then asked whether COP1 is involved. My previous kinetic analysis indicated that COP1 might be present during cisternal, I should say is present during cisternal maturation at the appropriate time to form vesicles for the intermediate pathway. As you can see at the top here, COP1 actually departs the Golgi well after the early pathway protein VRG4 leaves. And at the bottom, you can see a few traces highlighting the fact that the intermediate protein TMN1 actually departs no later than COP1. So this was promising, but we needed a functional test to determine whether COP1 actually forms the vesicles of the intermediate recycling pathway. So, and this actually proved tricky because COP1 is essential. We cannot simply delete it like we can with the AP1 and N5 adapters. So what I did was I rapidly and partially inactivated COP1 by attaching it to ribosomes, and I did this with the rapamycin-inducible heterodimerization system. For the assay, all I did was I added rapamycin, and after 4 to 7 minutes, I basically tracked cisterna over time that acquired a given protein of interest. And the rationale is that proteins that are dependent on fully functional COP1 for their departure from cisterna should experience an increase in their cisternal residence time when COP1 is partially inactivated. And this is actually what I saw for both the early protein BRG4 and the intermediate protein TMN1. Both proteins experienced a 1.5-fold increase in their cisternal residence time when I partially inactivated COP1. However, the late Golgi protein, which we know, KEX2, which we know to be AP1 and N5 dependent, did not experience a significant increase in its residence time. So this data directly provide evidence for dependency on COP1 by the intermediate trafficking pathway. And we believe that the intermediate pathway forms its vesicles with COP1. And we can sum this these data up then and fill in the missing information. So here we have 3 pathways, and we believe the first 2 pathways use COP1 to form their vesicles, and the last pathway uses clathrin and its AP1 and N5 adapters. But we didn't want to stop there because we also recognize that there are different proteins that are involved in the fusion step for each of these pathways. And we specifically wanted to find a way to differentiate mechanistically between the early and intermediate pathways, since they both apparently rely on COP1. So we actually started by looking at the Golgi family of vesicle tethers, because these have been characterized in mammalian cells by Sean Monroe. And The architecture of Golgins is relatively simple. They have C-terminal domains that associate with Golgi cisterna, central regions with multiple coiled domains that provide length and a degree of flexibility to the protein, and N-terminal regions that have been demonstrated in mammalian cells to have amino acid sequences that are thought to contact and tether vesicles. So these Golgi tethers are thought to initiate the vesicle fusion process by tethering vesicles at some distance from the membrane and then helping to localize them closer to the membrane where components of the downstream fusion machinery can better engage the vesicle. So to begin our analysis of budding yeast Golgins, we performed kinetic mapping for 3 of them, and we found that this mapping suggested that each Golgi participated in a distinct intra-Golgi transport pathway. For example, the Golgi RUD3 here arrives shortly before early pathway proteins during cisternal maturation. Interestingly, we observed that RUD3 departs only after these early pathway proteins have reached their maximum levels, suggesting that RUD3 is present during the fusion stage for vesicles of the early pathway. And we made similar observations for the Golgins SGM1 and IMH1. SGM1 arrives shortly before intermediate pathway proteins, and IMH1 arrives shortly before late pathway proteins. So these data collectively suggest that RUD3 may be a specific Golgi tether for early pathway vesicles, SGM1 for intermediate pathway vesicles, and IMH1 for late pathway vesicles. So to test whether or not these Golgins can actually function as vesicle tethers for these specific trafficking pathways, we devised an ectopic vesicle tethering assay. And essentially to do this, we modified our vesicle capture assay that I presented before. So now we're ectopically localizing Golgins to the septin ring, and we're looking for evidence of vesicle tethering. In the context of this assay, if we see a GFP-tagged transmembrane resident Golgi protein appear at the septin ring when a Golgi has been relocalized there, then we can conclude that that protein has been tethered by the Golgi in vesicles. So to begin, we looked at the Golgi IMH1, which we had hypothesized was a specific Golgi for the late pathway. And what we did observe is that when we localize IMH1 to the septin ring, the late pathway cargoes KEX2 and sterol 13 appear there. However, the early and intermediate cargoes that we assay are largely absent by comparison. So this data provides evidence that IMH1 serves as a specific Golgi tether for the late trafficking pathway, which is also consistent with our kinetic observations. We next tested the function of the SGM1 Golgi, and we thought based on kinetics that it was the specific intermediate pathway tether, and it turns out that the tethering data also support this hypothesis. I hope you can appreciate that the 2 intermediate proteins that we assayed do appear at the septin ring when we relocalize SGM1 there, whereas the early and late Golgi proteins that we assayed do not. Now, this tethering activity, I should note, by SGM1 is markedly weaker than that of IMH1 for reasons we don't understand. However, it is still very specific. The Golgi RUD3 now, we thought perhaps could be the tether for the early pathway based on kinetics, and our data, similar to the other 2 Golgins, also supports our hypothesis. And here you can appreciate that the 2 early Golgi proteins that we previously assayed using vesicle capture also appear at the septin ring when we relocalize RUD3 there. However, the intermediate and late proteins that we've assayed do not. So these data suggest to us that RUD3 does serve as a specific Golgi tether for the early trafficking pathway. So now we can sum up the results from all these experiments and add information to our master kinetic diagram. What we believe we've done is identified 3 molecularly distinct intra-Golgi transport pathways that operate at early, intermediate, and late stages of cisternal maturation. The first 2 pathways are dependent on COP1 to form their vesicles, whereas the last pathway, the late pathway, uses the clathrin coat and its AP1 and N5 adapters. Each pathway also uses a different Golgi tether to initiate the fusion process for its vesicles. The early pathway utilizes RUD3, the intermediate pathway SGM1, and the late pathway IMH1. I hope you can appreciate how each of these pathways has a distinct set of molecular machinery to operate it. And I hope you can also recall from the introduction since Golgi polarization is evolutionarily conserved, that these different these different sets of molecular machinery can be thought to actually explain mechanistically how this polarization occurs, at least for transmembrane resident Golgi proteins. Each of these pathways operates sequentially and is distinct because it uses a distinct set of machinery. In the future, we can further elaborate on these different definitions for each pathway by investigating the multi-subunit tethers and the snare complexes that act in each of the pathways. Finally, I want to thank the members of my lab, both Krahn and Forma, whose assistance was critical for the work that I just shared. And I also want to acknowledge our sources of financial and technical support, and thank all of you for your time and attention. And now I'd be happy to answer your questions.